So what happens now is we have approximately an hour and a half for the voting discussion and the votes themselves. And again, to kind of help inform the discussion, we have the patient representatives, we have our clinical experts, we have the ICER research team with the University of Colorado collaborator. And I thought I should take the opportunity, since Dr. Powell, you didn't get a chance to introduce yourself earlier this morning, and also just if you could say if you have any financial or other conflicts to disclose.
Matt Powell, I'm the director of the Division of GYN Oncology here at Washington University in St. Louis. I'm really honored to be a part of this process. I came in kind of late to the process, so I think I'm filling in for somebody probably. I do have lots of conflicts. In order to do research in this country, you need to be embedded, I think, a little bit with our pharmaceutical group. So I have, thankfully, conflicts with all of them. So there's really, hopefully that's not a bias, but certainly Roche, Genentech, Tesaro. I don't know if is it published somewhere in my conflicts, but anyway.
We have it on file.
You should have all
we have it on file, but it's not on the board.
It's kind of the nature of the beast as a researcher. I do a lot of clinical research and basic science research, and but I think you've probably heard I'm pretty passionate about trying to get new therapies for our patients.
All right, thank you very much. Okay, council, so let's get, first before we even start the discussion around voting, let's just kind of check our technology. So we are here in St. Louis, and we're going to have a test question for the voting technology. The question is, St. Louis is the first American city to host what event? The World Series, the Olympics, the World Fair, or the World Cup? And if the audience can see it as I can, you'll see the responses in the bottom. We should have 9 votes, so as soon as we do.
My screen doesn't say it was green. It says polling closed.
Yeah, same for mine.
Just reset. I'm going out and coming back in.
Do we
That's
need
because you
to
didn't answer
submit
correctly.
or just...
Okay.
That's okay, it just takes a screenshot, so.
So we don't need to submit the answer, just touch it.
If it's blue,
Oh, okay.
yeah.
All right, so we have all of our answers. You've all made your guess. 5 said, it's hard to see, 5 said fair. Can I move that bar?
Okay.
The answer is the Olympics.
Wow, that's right. First American city to host the Olympics. That's right. What year?
The Olympics, 1904.
1904.
I know this because WashU's football stadium is an Olympic stadium from 1904. It is very small. That is the only reason I know it.
So see what you get out of ICER meetings. You get history, you
Olympic
get
city.
culture.
Welcome to an Olympic city.
There you go. Who won the 100-meter dash in 1904?
That's great.
That's not the next question. Okay, so the council has the list of questions, and we will go through them one at a time. So the first question reads, in patients with recurrent BRCA-mutated disease, is the evidence adequate to demonstrate that the net health benefit of treatment with olaparib is greater than that of treatment with standard chemotherapy? Now, this will require you to think back to the specifics, and I will invite the ICER team to remind you of where this would be in the evidence summary. It will involve us in a discussion upcoming around the strength of evidence related to the types of studies, the types of comparisons that are possible, and we'll take it from there. So, Dan or Lipika, could you please remind the council where they can find what you think are the key data points on this in their evidence summary, and also just briefly, very briefly summarize what is in the ICER report that is specific to this question.
So in the evidence summary, the recurrent BRCA-mutated disease data for olaparib is in Table 3. And so a reminder that in this population, this was one single-arm trial. PFS of about 17 months and progression-free survival of approximately 7 months on a median basis. A greater improvement in PFS in patients who are platinum sensitive versus platinum resistant. And quality of life data were not reported in this study.
Okay. All right. So let me just, while you guys are looking at things, sometimes I'll ask some questions too. So as Dan just said, subgroup analysis of one single arm trial. This drug was approved for this indication 3 years ago, 2.5 years ago. So again, from the clinical expert's perspective, this is, however, the subgroup analysis does have overall survival data. So in a sense, it stands out in that way, but it also, as a subgroup analysis of a single arm trial, the evidence purists would say, where's the RCT? Where are the additional trials that would confirm this finding? The PFS of 7 versus 4 to 6 for a standard historical outcome doesn't seem like a great benefit. We did seem to get more kind of spread, if you will, on the overall survival. So can you put some of these issues around the strength of evidence and the findings between PFS and OS in perspective again from what you see? Go ahead, please.
So I think in terms of looking at the germline BRCA subgroup, I think it's also important to remember that this is a new therapy. The side effect profile is low compared to what we would alternatively offer them. And so I do think it's a very reasonable treatment choice for our patients. Even if there is no significant overall survival benefit, I think it's also hard at this point to confirm that, and also hard because these patients will go on to multiple other therapies. So it makes it very difficult to assess overall survival for this group.
Dr. Bell, everything?
I guess the only thing I would add is I know several of our advocates were saying we want more data, we want more data, we want to wait till the overall survival data matures. Unfortunately, we may not ever get that high level of data that's meaningful. I think is the one gentleman from, I think it was Tesaro, said, would require several thousand patient trial to show us overall survival benefit for patients that live such a long time and have such varying therapies throughout the point. So although we'd all love high levels of evidence, we may not get it because there's a lot of crossover. So patients that were on placebo, these real patients are all motivated to be on a clinical trial, right? So they're also motivated to look for the next best thing when the next best thing comes out. So I'm not surprised when we don't see big signals of overall survival, even when there's a very active drug out there, because of crossover. So my thought and opinion would be, although the low level of evidence and the number of trials is small to support this statement, it's in line with our clinical experience.
Yes, Scott.
So should we adjust our thresholds for what adequate evidence is in these populations? We sort of are rigid in our interpretation of a single-arm study of 140 patients. I think if you asked most rational people, they'd say, that's not even a study, right? It's just a collection of data.
I think the other thing is we talk in medians because it's easy to put in a table. But when we look at these long tails on these survival curves, we see something happening that's unique, where we have a lot of patients doing well for a long time. And this, as I mentioned, the duration of benefit really seems what drives it for me as a clinician for the PARP inhibitors. Once you find somebody who benefit, they tend to benefit for a long time. So it's a little bit different than cisplatin chemotherapy, where you get a nice response rate, you see how they do. They get too toxic to stay on it. They can't handle the cumulative toxicity of the drug, so they have to come off. That's why most chemotherapy is a 6-cycle type thing, and then you're done and you see what happens. That's not how it is with an oral agent. They'll be on for a long time if they're benefiting. So when you look at medians, you miss out for this tail approach on their survival curves that I think really is what drove approval, because some patients, or a large group of patients, did very well for a long time. And I think, so it's a little different interpretation, and just purely looking at medians, you get a little misled.
But when you talk about that tail, what percentage of patients are making up that tail? Because if it was a large percentage of your patients, then it would be impacting the median.
Correct,
So-
but
so
it
it's
does, well, no, it depends on where that median falls, right? So the median, if curves tend to do, if you're differentiating two groups right near the median, you can see large differences in progression-free survival without impacting the hazard ratio as much, right? But we're seeing huge effects in hazard ratio, at least on the maintenance side, when we have a comparator. We don't have it so much because we haven't compared it directly to chemotherapy as well. And that's the hard part. There's not as higher level of evidence as we'd like in this situation. I mean, we'd love to have more comparators directly to chemotherapy to help inform us on these decisions, but I do think that the real key in this is that when you identify the patients that do well, they do well for a long time.
Can I ask you another, just add-on question to that? And maybe I'm not knowing the evidence base as well as folks here. If we're talking about these kinds of distributional effects, why is it that that hasn't appeared in the peer-reviewed literature, the mean as opposed to the median, the distributional effects? Because it seems like that's part of the crux of interest in patients and clinicians, and yet it's not there.
I'm probably asking one of the statisticians to comment on the mean versus median issue, but you know, the hard part, I guess, also is these trials were designed, really before we knew what the right patient population was, who they were going to work in, how best to treat them. Did you treat it with low volume disease? You treat them with measurable disease? We're still mid in that discovery phase, so echoing the comments of our patients, it's there's still a lot that we need to know yet. But I don't know. Do you have a comment on the mean versus median?
I have no comment on that, but in terms of kind of looking at the patient population, in the clinical trials, third or fourth, or maybe even further line therapy history or germline BRCA mutation, as we're learning more about how PARP inhibitors work, we know that over time, someone who has a germline BRCA mutation who's been treated multiple times can have a reversion mutation or a mutation in that pathway, which renders them no longer sensitive. So you're getting this mixed population in the clinical trials, whereas we're learning more how to treat them, and we know, and like you were mentioning, we get a feel of who we think is going to respond and who's not based on how did they respond to their prior platinum treatment, how long was their disease-free interval, and as physicians, we can kind of already predict who most likely will respond well, and that's not really well shown in the initial trials, because those initial trials, it wasn't perfectly clear that that's kind of a signature of who's going to respond.
And some people have had 8 prior lines of therapy in the initial trial.
And so it's not
a
surprising that
mixed
they
bag.
wouldn't respond.
Okay. All right. Council, any other questions about this particular vote? Yes, Don.
Well, so I live in a world of evaluating evidence, grading it, using methodologies, things of this sort. So, part of the challenge here is in this question, what do we mean by adequate, and what do we mean by net health benefit in that context? So the way I'm viewing this is that there is, even if it's an observational study, evidence, okay? It's not a randomized control trial, but it is evidence. So there is evidence. Is it adequate? The net effect, is, it seems, on a median basis, small at best, and the numbers are low from the standpoint of making general conclusions. And so I'm trying to live in the world of strength of evidence and grading, and it would seem as though if I were applying some method, it would be weak at best, and maybe more inadequate. So I'm just trying to figure out in my mind how to balance what I see here in that context. I guess the question would be, is this, and I don't know if this is an appropriate question for clinical experts, but is this something that oncologists are recommending at this point in terms of treating patients with this?
Yes, definitely.
Yeah.
And patients are demanding it, honestly.
Yeah, right.
I will.
That's not surprising.
And I would say, Don, that your comment about the level of evidence, I think a lot of us were shocked that all comers got approval for niraparib and olaparib based on pretty small sets, with the delta being not nearly as impressive as what we're talking about here, with the germline patients having these benefits. And so it has clouded the issue, and it makes it hard to make these broad statements. But I still completely agree with what you're saying.
And obviously, you want to do it in the context of shared decision-making to be sure patients don't have overinflated expectations, so I get that, but that's helpful, thank you.
The other thing, and as you think about it, as you laid it out, I would also encourage you to think about, remember, net health benefit includes the harms. So we can talk about the strength of evidence for the PFS and OS. If you think about the harms as a part of this equation, I think obviously that's a key part of the net health benefit. Any other questions from the council?
I think one point on harms is good. We now have some data to say when we give a PARP inhibitor, we haven't hurt what they can get next. They still seem to respond to the next line of chemotherapy quite well. But if I give another cycle or a round of carboplatin, I might have ruined their bone marrow enough to where I can't give them something else. So there's some hint that we maybe don't have as much harm with the PARP inhibitor giving it now than we would giving another round of platinum. So I don't putting that in context, but it's hard to point you to a clinical trial that would prove that.
Right. I was going to say, I want to come back to that as part of the policy roundtable, because I think that's a good point to bring that up then. All right, so with the number of, I'm sorry, Shumei.
I think somebody might have asked the questions in the, the call. It's about the progression-free survival and the difference between what's in the trial and in the literature kind of is not quite different, but the overall survival kind of is 16.6 versus 8.9. It's quite different. And it's also mentioned because in the trial for the overall survival is based on the 193 patients, which is about 18% of those who had 1 or 2 prior chemotherapy. That's probably too detailed. What is the potential impact of that? 18% have 1 or 2 prior therapy on that overall survival. It's probably too detailed.
Well, I'm not sure I'm following your question. I know on one call we talked about the slightly unusual finding that these using historical comparisons, which is obviously fraught with difficulty, we seem to get more spread on the overall survival than on the progression-free survival. And it's usually the other way around. You usually find a longer progression-free survival that may not pan out in as many months of overall survival. So again, this was brought up on the clinical expert call, but if you have any additional comments.
And also in that longer overall survival, part of the reason is because some of those patients only had 1 or 2 prior chemotherapy. What is the potential impact of that factor on the longer overall?
Yeah, so part of the tricky part is we're dealing with with BRCA patients, which is not most of the early trials were enriched for BRCA positive patients, and that's what the question we have at hand. Those patients not only tend to respond well to PARP inhibitors, but they tend to respond better to all chemotherapies. So a little bit your question about the big, small PFS differences, yet OS differences, has a little bit to do with a lot of other therapies working quite well in this population. So there's some uncoupling of what happens from PFS to OS, probably more so than any other subset of ovarian cancer, because of that opportunity to respond to multiple different agents. And I don't know if that's directly answering your question, because I'm not sure I completely understand your question.
But you're raising another missing piece of data. It's missing because it doesn't exist. Does somebody who's had just 2 prior courses of chemo do better on olaparib or a PARP inhibitor than somebody who's had 6?
We do have some response rate data that would suggest those with fewer lines with a mutation are going to have a better response rate. when they get the drug than if they had 5 or 6 lines of therapy. So there's a hint there, but people do tend to respond or not, too. So when you're that patient in your own test tube, you can see what happens.
I know.
Just to be
15
clear,
months,
though.
I had six courses of chemo before I started this, so.
Just to be clear, since you guys are needing to vote on the evidence we presented, we presented the evidence that links to the FDA indication for olaparib in this population, which is the subgroup analysis of patients who got olaparib as a 4th line or later treatment. So they had 3 or more prior lines of therapy.
Okay, so I know that some of you have already voted. As we go forward, I always think I should suggest that you hold your votes until the conversation is done, because you may always, there's a risk that you'll hear something that you'll want to use to change your vote. But for the rest of you who have not voted, or if you want to re-vote, you can always just update your vote. But let's go ahead and have the votes. So we're waiting for one more. Okay. All right, so 5 said no. The evidence is not adequate, and 4 said yes. All right, because of the other votes that we've got, we're going to need to keep moving forward at this point. So the next question is, in patients with platinum-sensitive disease who are eligible for maintenance therapy, so we're shifting clinical domains now, or clinical indications certainly, is the evidence adequate to demonstrate that the net health benefit of treatment with olaparib is greater than that of surveillance alone? Lipika or Dan, you want to summarize again the availability of evidence in the ICER rating?
So page 8, table 4 is the summary of olaparib for maintenance therapy. So there are 2 studies, phase 2 study, which is known as study 19, and a phase 3 study, SOLO 2. Some differences in those studies, as Lipika pointed out, both in terms of the dosing and formulation as well as in the patient population. So study 19 was a mixed population with retrospective identification of BRCA mutations. I think a third of them had BRCA, 36%. SOLO 2 was nearly all germline BRCA. And you see the results for both studies laid out in that table. A larger benefit on progression-free survival with women with germline BRCA mutations than without, and some overall survival data, but immature at this point.
Questions from the council?
Oh, sorry, one other thing, because I know Dr. Warner Hendrickson pointed this out earlier. So no differences in, the quality of life was measured, no differences between olaparib and placebo, which can be viewed, depending on your opinion, as a benefit, given that the comparator is observation alone.
Just invite one of the clinical experts again to please, when we hear no overall survival shown, can you just comment again on the maturity of those data, the crossover issues, just how to put that in context?
Again, when it's not a primary endpoint of the trial, it's not powered to show that, and there would be no expectation that you would necessarily see that in a trial of this size. If you did, you would know that, if you do see a survival benefit, you go, wow, that drug must be really working. But given the level of benefits of these drugs, we would not be under the expectation that we would see an overall survival difference in a trial such as this size.
Okay. So we've got progression-free survival, we've got 8 versus 5, is that right, from the table? This population. For the overall population.
So it depends on the study. So in SOLO 2, it was 19.1 versus 5.5. That's again all germline BRCA. In study 19, it was a
germline BRCA. Are we looking at the same thing?
Looking at the second row.
Okay.
SOLO 2, PFS 19.1 versus 5.5 for placebo. For study 19, it was For BRCA mutated, 11.2 versus 4.3. And for BRCA wild type, it was 7.4 versus 5.5.
Can I ask a follow-up question about this?
Yes.
So the call of the question and the indication itself is not limited to BRCA mutated, germline BRCA mutations, but the trial itself was, as you said, 97% of patients fit that criterion. So if you had advice for us on how to interpret the patient population in the study as it compares to the more varied patient population in study 19 and we have in the question, anything would be helpful.
Platinum sensitive disease.
Okay, but it's not eligible for maintenance.
Because
Yeah,
the indication is drawn more broadly.
The indication is drawn more
Yeah.
broadly. The question is worded more broadly than germline BRCA versus not. So I think it's, in your opinion, in your judgment, based on where the data fall out, how you would think about it with this question. It's not much more I can say.
Why is the question that?
It matches the indication. So the FDA indication is for both germline BRCA and non-germline BRCA mutations.
If we want, we can split this
I think
question.
you have to say it matches the current indication. The time you guys put this data together, the indication was different. And with just G-BRCA at the time this data set was put together. Because you haven't included all the other updated data, you're just including the updated FDA label when you're interpreting this question.
So when we started the project, there was no indication for olaparib in maintenance therapy. That came during the course of the project. And so we had data from both studies, although the Solo 2 publication didn't come out until later in our process. So we actually focused attention on germline BRCA in our earlier drafts of the report, and then the FDA indication came in more broadly.
Okay. That's true.
So I guess back to Rachel's question, do we need to split this voting question to make sense of it or not? Is the data?
To be clear, I'm not necessarily proposing
No,
that
I
we
know.
do so. I
You
just
want a
find it
clarification.
challenging.
So, I mean, Dan, you know, and Lipika, you guys know these data better than I, so we need your guidance on how to do this vote.
I mean, we know that the prevalence of testing varies widely across the country. So by splitting the question, you might actually in a way be saying that if people aren't able to get the testing, that they shouldn't get the treatment. So that's, I don't know, that's part of what we have to think about. That's not necessarily an answer.
But that's helpful, thank you.
Yeah, I think that the data are here. You can see that the performance is different depending on whether there's a germline BRCA mutation. I guess it's up to whether the panel feels more comfortable with two different questions versus one.
It's up to us.
It's up to us. I think that if generally what we're trying to do is to use the FDA indications as a guide for evaluating the evidence, that we should stick with that.
Okay. We can have notes are kept with the votes, so that the vote can note that the panel wants to distinguish in some sense or bring to people's attention the distinction in the type of evidence available between the two different subpopulations, but the vote will represent a general judgment across both. Any other questions? Yes.
Possible make a comment?
Yes.
So I would say the FDA, I don't know the full inner workings of the FDA, I'll put that on the line, but they approved all comers not because of this study. They approved all comers for olaparib because they approved it for niraparib. The interpretation basically was these drugs are all about the same. Let's make the approval about the same. There's no way on this little data set by itself it would have made approval. So in reality, the approval that olaparib have is based on G-BRCA status. And this clouds it here, this non-germline. So the weight of the evidence here is all in the G-BRCA group.
We could have a whole nother afternoon about why the FDA decided to do that. When in checkpoint inhibitors, you can't always assume that it's the same outcome.
If you use olaparib data in and of itself, there's no way you can feel good about the non-G-BRCA population. Well, that's a pretty strong statement.
All right, so we have now, right? We've now left the council with a firm grasp of how challenging this vote is. But why don't we go ahead and vote, since I want to be sensitive to make sure we have time for the other votes as well. All right, thank you. Okay, 7 yes and 2 no. I'm going to try to take a brief opportunity, maybe should have done this in the first question too, but I'm going to try to weave in a chance. Can I get one respondent or one voter who voted yes to give their rationale, and then one of the votes for no to give their rationale? So why was this a yes? Anybody willing to give their rationale who voted yes?
We see a,
I was gonna weigh in on, I think the quantitative data is related to progression-free survival, and then I think a judgment on this idea of, eligible, like what was the eligible for maintenance therapy, and thinking about that in terms of resolving the issue of the BRCA mutation or not. It's kind of how I decided that I didn't have to put much weight on the other dimension of the data where it was not as strong.
I'm wondering if part of the reason of the flip, if you will, the other was 5 no and 4 yes, this 7 yes, 2 no. Were people viewing the role of there being RCTs here, or 2 RCTs? Was that a part of people's thinking? I'm seeing some nodding heads.
yeah,
She might-
the first one I voted no, this one I voted yes, because look at the progression-free survival, this one, the difference is pretty big. And the first one is very similar based on the other, the literature, the comparison with the literature. Although the previous one have a large overall survival, but I question it because this is not in the summary, but in the full report. When you estimated the overall survival, you have 18% of people have the first line or second line treatment, and that's potentially a fact to lead to the longer overall survival. So that's why I didn't vote yes for the first one, but vote yes for this one. Because I questioned the first overall survival based on that 18%.
Any comments from someone who voted no on this one to help distinguish their reasoning? There are 2 of you who voted no. Yes, Jill.
I voted no because I feel like if there's anything about all of today is the evidence isn't adequate. If all comers can take olaparib, and the difference between, and granted we do have 2 randomized controlled trials, with 36, we don't get to pick, or I guess the maintenance therapy is given to all comers. So if you're 36% of your population is germline, and so 64% isn't, I guess at best you've got an overall survival benefit of 4.4 months, which is typical with cancer trials, a progression-free survival difference of 7 months. And whether or not that's statistically predictable, because you've only got like 60, 70 people per trial, or per arm. I would like to see adequate evidence. I didn't feel like it was adequate.
I'm the other no, and I agree with that. I think the fact that this is a pooled trial
Sorry,
data.
Rena, can you pull this just a little closer to you?
Sure,
So
the
we-
fact that this is a pooled data analysis, but we know that there's a subpopulation that likely is the responder here, or the more likely responder, suggests to me that the company, it behooves the company and/or the FDA to reconsider the label.
All right. So let's keep going. The next question, we are shifting drugs, going back in a sense away from maintenance back to treatment of recurrent disease. So in patients with recurrent platinum-sensitive germline BRCA mutated disease who are eligible, I'm sorry, this is another maintenance question. This is also, who are eligible for maintenance therapy, is the evidence adequate to demonstrate that the net health benefit of treatment with niraparib is greater than that of surveillance alone? Vivek and Dan?
Yeah, so I'll need to add some nuance in here. So we don't have, so the trial of niraparib in recurrent BRCA-mutated disease is still ongoing. None of those results have read out. I'm looking at the manufacturer for confirmation, right? So the Quadra trial is still ongoing. I know, I'm just trying to tell them why there's no recurrent BRCA question. Okay. In maintenance, unlike the previous question, we actually have two questions, one for germline BRCA and one for non-germline BRCA. Niraparib also has a broad FDA label, but the trial was designed to separately recruit and randomize women with germline BRCA and women without germline BRCA. So it was powered actually to detect differences in both subpopulations, so we're asking the question about these subpopulations separately. And so you see the results for both germline BRCA and non-germline BRCA on page 10 in table 6. OS data are still immature. PFS median for germline BRCA 21 months versus 5.5 months for the comparator. For non-germline BRCA overall, 9.3 months versus 3.9 months for the comparator, placebo. You also see some subgroups defined by HRD status as well as other types of BRCA mutations, somatic or wild type. And so, and you see the results there. And then for HRD negative, without any BRCA mutation, about 7 months versus 4 months. So I guess the important numbers would be the top lines for PFS across these two populations.
From an RCT of how many patients?
I have to try to count this up here. So 553? Okay.
Okay, questions from the council about this study, this evidence base? Patients or clinical experts, anything that stands out about this evidence that is of note? We've already covered some of the key issues.
Yeah, again, it just points us to the direction of the patients who seem to benefit most, but there did appear to be benefit across the entire trial. And it had the larger subset of the non-HRD, non-GBRCA population, showing still a benefit, although modest compared to the HRD or BRCA positive patients. So,
Can I just ask
Sure.
a question? It's not directly relevant to answering this question, but olaparib and niraparib got the maintenance indications roughly at the same time, yet this trial was designed to separate these subgroups and the other one was not. And do we know why that was? Do we have any information about that? I'm just assuming they were conducted roughly at the same time. So it's not
was
as if,
much earlier. We had really no idea it would work in wild-type patients at the time.
but then the approval came at later, is what I'm suggesting. So that trial you're saying was actually conducted years earlier?
So if you look at these trials,
Yeah.
the olaparib trials happened earlier, the niraparib trial more recently,
Okay.
but that, there was some overlap in that, but the
Right. That's what I'm suggesting.
the niraparib trial was specifically designed for maintenance, didn't have a treatment group. It was an earlier experience with olaparib.
Yeah, I'm
and this-
focused on the maintenance indications here,
Focusing on
which
the
came 3 years
maintenance
later.
indication. So
Yes.
this design of comparing it to placebo came later, and it's cleaner. We like the data. It's easier for us to interpret when we look at it. But it's again, we're not comparing it to chemotherapy, we're comparing it to placebo.
No, I understand that. I'm asking a different question that I'm not sure I've received an answer to, and there may not be an answer to it that we know, which is
Why did they get the approval at the same time as, or roughly the same time, I think was your question.
I'm suggesting that the trials were conducted around the same time, and if so, why is one trial designed in a way that enables us to answer this question more effectively than the other one?
I don't know if this helps, but so olaparib, the studies were started a little bit earlier, if I recall correctly, and initially they went into the FDA, and correct me if I'm wrong, with the maintenance implication, and that was initially declined, and then they went into a second. And so I think olaparib had started in terms of getting some of the data, so like the study 19 is just an older study before we were really aware of exactly who's going to respond best. And from that study, that's kind of where some of that information came about, okay, it's, it seems like these BRCA mutated patients are doing best, and from there.
And probably the real
That's very helpful.
backstory,
Thank you, Doctor.
backstory is we were worried about myelodysplasia and AML at the time. So maintenance was felt to be too risky to put the patients on a maintenance trial or to give an approval for maintenance based on that risk. Now, the risk for AML and myelodysplasias remained low, and so people had more comfort recommending a maintenance type strategy. People, I guess there was 3 hematologists on the initial FDA approval panel for olaparib that really were not comfortable with that maintenance indication. So it probably more was following these patients over time and not seeing that toxicity signal that led to the maintenance indication rather than any change in the data. I may be wrong about that.
So, and based on the testimony we heard earlier, it was at about the toxicity in terms of fatal toxicity, it approaches 2%, 1.5 to 2%. I'm kind of looking over there to get my facts correct. Does that sound like I'm in the ballpark? Okay. The other thing I just wanted to highlight, at least if Susan and Betsy want to chime in and add some color to the fact that we do not see a quality of life benefit, but more specifically, we don't see a decline in quality of life of patients that are on maintenance therapy. If you have any kind of comments that I mean, I think that's an important consideration in all the things we're considering today, and I just want to make sure we don't lose sight of that one.
Oh, well, the only thing that comes to mind is that I wonder if they looked at the quality of life index when the patient was first put on it or when the dose was adjusted to make it more as effective but fewer side effects. I don't know if that information would help.
Yeah, so when they looked at quality of life, they looked over time, and so there really was no significant health-related impact kind of in their quality of life while they were on therapy, which if you're comparing to placebo, it would be very hard to see a benefit in quality of life. I think the key is you want to make sure there's not a significant, we're not decreasing their quality of life, and I think at the ASCO session, they were then doing other...
Time without symptoms.
Yeah, time without symptoms, and because that is longer, are you possibly, improving their quality of life because they have a longer period of time without symptoms, which, they kind of stated that's actually an increase in quality of life, but that's harder to calculate.
Yeah,
I would say the other tricky part is the group that was on placebo that progressed, they came off trial, so they no longer were in the comparator group. They went on chemotherapy. Their life got a lot worse when they went on chemotherapy, but we're still comparing to just the placebo group.
Right.
So the way we design our trials isn't perfect, but I think you're right to feel they're probably equivalent whether you're on placebo or pill. But we hope we're actually staying on an agent keeps you off chemotherapy, which I think we'd all agree is clearly worse than being on.
Yeah, and I'm not looking at the language in the evidence report at the moment, but not that this is a time to add edits, but as you think about how to characterize this, I think it's a little bit different to say that we don't see a benefit in quality of life versus saying we don't see a decline, right? And so I think
there's newer publications that are out now too.
I think it's a better way to characterize the trajectory of quality of life in these trials rather than it suggests that we're expecting a benefit when in fact we're just expecting, ideally we would see no difference.
Yeah. Okay. But also recognize that your comparator group is just the placebo group.
Yes, right, that's really important.
It does fall off when they go on chemo,
yep.
So it's not the data set. I think if you did have them stay on their tox assessments, you would probably see that difference where they may do better when they're on their PARP inhibitor.
So can I just make sure I understand what you said? So the placebo group is, the quality of life is only while they're in progression-free survival.
Right, once they progress,
Once
they come
they-
off trial and no longer do the tox assessments. So it's kind of a problem with the way we do our studies sometimes. It is a problem. We're changing that. We're getting smarter, but we're not very smart.
All right, I'm going to need to move us forward on this vote because of the importance of some of the latter votes we have as well. So please go ahead and vote on this question.
I think that's, they came off tox assessments once they progressed.
So 8 yes and 1 no, very similar to the previous question. So I think we can spare the interrogation of the yeses and the single no. Let's go forward to question 4. In patients with recurrent platinum-sensitive disease who are eligible for maintenance therapy and do not have germline BRCA mutations, is the evidence adequate to demonstrate that the net health benefit of treatment with niraparib is greater than that of surveillance alone? Same drug. I feel like we've had the conversation about the two populations. You can look again at the delta, if you will, between the progression-free survival. We've talked about the study. Does anybody have a specific additional question? And maybe this one doesn't stir up any additional.
I just want to clarify,
Sure,
in this
sure.
population, it's not expected that these patients would do as well, correct? In the wild type, is that what was said earlier?
Again, they're
Regardless
also
of what
including
they
the
get.
HRD positive and the somatic BRCA within this question. So you can't really look at that table and get your answer. Is the hard part about this question, because there's still the somatic BRCA patient population, which is about 7% of all ovarian cancer patients will have it, or 5 or 7 will have a somatic BRCA mutation. And this HRD positive group, which is basically the group that has the same broken machinery as those with a BRCA gene. So with HRD positivity, at least in this population, made up a pretty good part of the patients enrolled in the trial. So this question, I don't know if you're just trying to get at the all negatives or really the 3 groups. They're kind of these, the way they did this trial is, it's been an analogy as Russian dolls. You have the whole population and you have one doll inside and then another doll inside as they did these different populations and the way they did the data analysis. I'm just making it more confusing by bringing that up next.
So all we were trying to do is to call out the subgroups that were described in the study publication and other presentations. So, so you already voted on germline BRCA. So then within non-germline BRCA, there's an overall number, and that's the PFS you see, 9.3 versus 3.9. And then within that, there were reported subgroups by HRD positivity and whether it was somatic BRCA or wild type, and then HRD negativity. So those are the results you see in that same table 6. But they're all part of one whole, which is non-germline BRCA.
I have a clarification question regarding the risk of AML and MDS. Is it that there is truly no evidence to suggest that these patients on this treatment are developing those conditions, or that technology to address AML and MDS has arrived so that if it's a risk, it's a less serious one?
You want to take that?
Well, no, I mean, I think there is a known risk of AML, MDS is low, but I think it's also important to remember that, any DNA damaging agent like carboplatin also does have that risk as well. So it's also hard to tease out, because all of these patients have had platinum before, would they have developed it anyway? I mean, we don't know, but there is that associated risk. But again, unfortunately, the regimens that we have for our patients now, there is that risk just with their frontline therapy.
And did our...
I don't know if you want to add.
I think the risk has remained small. It's there, but it's near the background what we would expect for people that have 5, 6, 7 lines of therapy.
Mm-hmm.
So, I mean, it happens with all chemotherapy. We were worried that it might happen a lot more frequently with a PARP inhibitor, just the way it affects the bone marrow, but that hasn't panned out, or at least at this point.
So in our incremental effectiveness ratio, we did not include additional treatment for the AML or MDS, because essentially the risk is the same between the groups, correct? Okay, thank you.
Well, I think even more to the point, we focused on adverse events of a certain severity that occurred in more than 5% of patients, which was, I think, never the case in any of these trials for that particular adverse event.
I see. That's very helpful. Thank you.
But it's such a major safety signal that did drive some of the approval process for this.
I know I read this somewhere, but could you verify, this is a simple question, I think the overall survival for HRD negative is lower than HRD positive. Is that?
So the overall survival is not mature for this study. We don't have that data, at least that
Okay.
I'm aware of.
All right.
The data set for overall survival is not mature.
But Don, are you asking generally if
Generally,
the
yes.
prognosis is
Yes,
poor,
yes,
more poor
yes.
for?
Generally.
So in general, HRD negative patients do worse, not only with PARP inhibitors, but really all chemotherapy. So an HRD is really a marker of chemosensitivity.
That's what I was
as well. So
asking.
you're exactly, I misunderstood.
That's okay. Thanks.
Okay. Any other questions? Not, let's go ahead and vote on this subpopulation, please. All right. 6 and 3. Okay. So we have slightly more people who said no on this one. Would someone who flipped their vote from yes to no care to explain why? Make that clear for the audience and the record? Or not? Somebody?
I didn't flip, but this was right on the edge for me, so I can see.
Okay. Perhaps a slightly smaller gap in the difference in PFS. Okay, moving to question 5. Different drug, single question. In patients with recurrent BRCA-mutated disease, is the evidence adequate to demonstrate that the net health benefit of treatment with rucaparib is greater than the treatment with standard chemotherapy? Flip again down, same drill.
Yeah, page 12, table 8, subgroup analyses for third line or later use from a single and possibly a pooled analysis from 2 single-arm studies. So you see the results for Ariel 2, which is a single-arm study, and a pooled analysis of patients with BRCA mutations from an earlier study and Ariel 2. Those are the 2 results that we reported. In terms of PFS, platinum, so in the Ariel 2 only analysis, about 13 months. In platinum-sensitive patients, about 7 months in platinum-resistant patients. And in the other study, about 10 months overall of PFS. And so at this point, we only have historical comparator data, and for second or third relapse with historical therapies, median PFS is about 6 months as a loose comparison.
Okay.
And then we neglected to mention that there's a harms table for each drug as well. So table 9 on page 13 reports the adverse event profile for rucaparib.
So the clinical experts have spoken before about modulating the dose to the side effects, because still, a large 100% of patients have some adverse event. But I also wanted you to comment, because I'm not sure we did it clearly enough before, but I just want to make sure. The use that ICER was left to, which was the approach of looking at a standard kind of outcome from the historical data with which to compare these progression-free survival numbers. From the clinical expert community perspective, is that 6 months of standard PFS? Does that kind of ring true? I know we've bounced that off of multiple clinical experts. But for the council, when they're trying to figure out what the net health benefit is, and they hear a PFS of 10, we've told them that the historical outcomes were around 6. Can you comment on that?
I think from a just clinical standpoint, the PFS of 12.7 months in platinum sensitive is great. I think 6 months is reasonable. I would say, though, patients who are on their third line or later that were platinum resistant, I would say oftentimes it's shorter than that.
Yeah, the data that's quoted is usually quoted for primary platinum sensitivity. That means that's the first time with platinum, how they did after that when they got treated again with a therapy. So that's their second platinum. As we recognize these drugs, some of these patients may have had 3 platinum regimens or more going into this, and so that number would skew slightly based on that, usually in the downward direction.
So what you're saying is that the clinical expert community sees a PFS of 12.7 or of 10, and they think that looks better than historical outcomes.
Yes.
Correct. But again, that's the difficulty of not having a direct comparative study. So these are 2 single-arm phase 2 trials, and I think Dan has kind of gone through that. Any questions from the council about the data on this drug? Yes. Brett.
From, I guess, a clinical standpoint, is there, what is the, as a clinician, choosing between olaparib and rucaparib? I mean, thank you.
Rucaparib and olaparib.
Yes. For this population, what goes into your decision making between those 2 drugs, or is?
Oops, can I actually? You must have been reading page 3 of my notes. I would love to come to that question in the policy roundtable.
Okay.
Because it's.
I will table that.
But it reminds us all that the reason we're not having that discussion now is because the evidence on the patients was so different in the studies that we did not feel that we could ask that question of you to vote. But we will come back to it when we talk about the clinical implications.
I get why you're asking me to hold that. At the same time, I'm like, if I voted one way on one, I'm just having a hard time.
No, I get it. I'll allow the question, as the judge would say.
No, we'll wait. It's okay.
No, if you want to hear about some of the distinctions and how it relates to the evidence.
I don't know if it'd help anyway.
No, but it is interesting, the quality of the data when you look at olaparib, when it was done earlier, when we didn't know as much, it's not as good a quality data, but we've essentially accumulated all this other data from 2 other drugs that are similar, as you're pointing out, Ryan. They're not that different. And you hold it to a different standard because of when the trial was done, how the trial was designed. We weren't as smart as we are. You don't have the pure data set. There's a lot of moving parts here.
Great. Any other questions about net health benefit for this drug? Okay, please go ahead and vote. Yes and 4 no. That is close to the very first vote that we took. Might as well try to grab a quick why yes and why no. Somebody who voted yes? Or maybe I'll start with the no. Sometimes no is usually easier to describe. Why did you feel the evidence wasn't adequate for rucaparib?
I mean, I struggle with, we have a pretty positive response compared to what, yes, I know 12 months is very good, but how do I know that giving some other therapy wouldn't give you 10 months in these patients? I know clinical experience from your standpoint, but from evaluating the evidence, again, as I said before, a single-arm study, it's just you're just aggregating data and not comparing it to anything.
The response rate of 52%, in these populations, is well above the bar we would set in our developmental therapeutics committee for the NIH, for the NCI. So it would be definitely meeting it. Yes, it's this exciting. You're being asked for quality of evidence and the level of evidence. I fully get the no vote because you didn't have a chemo arm, and that's I don't know if it's my even role to respond to that, but it's very true. I mean, but we have to try to weigh the evidence that we have in front of us to see But I would say clinically, yes, I'm excited about a 52% response rate.
Okay.
And
Yes, Rachel.
no, I also find it difficult to evaluate here when all we have is a single-arm trial. And just to make a note here, that all of these patients do have BRCA mutations, but they're of different kinds. And it may be, I just don't have a sense of whether even within those populations there's a differential response, and so whether the differential inclusion of those populations within the trial contributed to that response rate. I just don't have a sense of that. So.
Yeah, if you want something to look really good, you make it all in BRCA2 gene patients. They do the best on everything. No offense to anybody that's not BRCA2, but it's, but really, there's dramatic differences. You're going to do better than if you're somatic BRCA or if you're just HRD positive. So we can stratify, but again, it just identifies a population. It doesn't say what that individual patient will do.
Yeah.
Okay. So we are now going to move into a different domain of discussion and votes, for which our panel here is still very germane. So this is the section of voting on other benefits and then contextual considerations. So just to give a little bit of the inside baseball, some of the people in the meeting may not know this, but again, ICER has gone through a prolonged period of revision of its value framework methods with public input on several different occasions. And what we have always done is include this kind of conceptual idea of other benefits and had it as part of the discussion. We wanted, with input, to try to figure out how to make this even more tangible and explicit and to have some kind of vote specifically on each of these kind of different domains. And there was a lot of discussion about whether that could be somehow quantified and fed back into a cost-effectiveness model. The ultimate decision was to not try to do that, given that there are no kind of consensus methods for doing so, but that to learn from the experience of having votes and discussion and specific votes on these, and then making sure that that's a very clear and kind of important part of the final output of the report. So this question says, when compared to the pegylated liposomal doxorubicin and carboplatin dose, well, when compared to the, sorry, how about we say when we compared to other chemotherapy? Does olaparib, and here I'm going to pause just to say, for these votes, because these drugs all have the same mechanism of action, we're not trying to distinguish between olaparib and the other two. We're really looking at PARP inhibitors. So I could almost say, when compared to chemotherapy, do PARP inhibitors for recurrent BRCA mutated disease offer any of the following other benefits? Please select all that apply. And I'm going to read them now, and then we're going to spend some time talking about them. A, significant direct patient health benefits not adequately captured by the QALY. B, reduced complexity that will significantly improve patient outcomes. C, this intervention will reduce important health disparities, and the longer version says across racial, ethnic, gender, socioeconomic, or regional categories. D, will it significantly reduce caregiver or family burden, broader family? E, does it offer a novel mechanism of action or approach that will allow successful treatment of many patients who have failed other available treatments? And F, does it offer significant impact on improving return to work or overall productivity? And I'll also note that we have the possibility, if another other benefit has arisen in the discussion or comes up now, we can, at our discretion, add it as another dimension for voting. So let's handle these right now. I'd like to kind of invite the council to consider these, to engage with the patient representatives, perhaps in particular, the clinical experts, to think about these other benefits and how they should factor in, if you will, or their relevant importance to considerations of value here. Don?
And does this, does answering these take into account, Steve, or should take into account the scientific evidence as well as the testimony, as I'll call it, from our patient representatives as well? If the answer is yes, how do you balance that?
Okay, I think the simple answer is no. It's not to say that evidence isn't important for these. So for instance, if there were specific gathered evidence on productivity or on caregiver burden or these other things, we would put it in the report and we would want to highlight it here. Often, however, we won't have quote-unquote data, but we will have testimony. We will have experience, accumulated experience over years, and some of this will just be intuitive. So it's a mix of those. But this is not meant to have you reflect back on whether it was a single study or two RCTs. This is, in a sense, that's somewhat irrelevant here. Unless we were talking about data that was gathered in an RCT and we were trying to figure out if it was different or not. So let's just work through these. Again, you can start to take notes and you can think about which of these might seem to be most relevant for this particular comparison. And Dan?
And if helpful, our ICER's take on these considerations are on page 20 of your packet.
So while you guys are thinking, I can see you thinking. I'm going to invite our patients who have come from a distance. You look at this list, you think about what matters to patients when it comes to value. We've heard about some of these already today. So hit the nail again with that hammer or pick a different one and describe if you think that the PARP inhibitors offer specific other benefits that the council should recognize in its voting.
Do you mind if I made a comment while there's
Please.
So, the framework of this question, the way it's designed statistically, is to compare a chemo versus a PARP inhibitor. And it's not that simple. They're going to get both of these drugs eventually. You're just choosing what they're getting now right in front of you. You can give them carboplatin, liposomal doxorubicin right now, and they can get the PARP later. Or you can do the PARP first and give the chemo later.
Point.
Unless we take these drugs off the market, a G-BRCA patient's going to get both of these. We're just arguing about sequencing in reality and costs. So it's a little bit arbitrary to say when we're comparing this, because you're one snapshot in time.
Yeah, actually, that's extremely helpful. And again, this is our first time working through the voting mechanism on these other benefits, and even framing it as a comparison, you've made a very helpful distinction. I almost now want to change the question to almost saying the world with PARP inhibitors in them versus the world without PARP inhibitors.
I think that's a more fair assessment, because if you look at them as comparison,
it's not the right question.
It's not the right question.
Very helpful point. Yes,
Okay.
please.
Looking at this list, all of them are benefits, except the only one I would question has to do with the one on this intervention will reduce important health disparities, so forth and so on. And the reason I say that is PARP inhibitors are generally given in large academic centers. I happen to come from a rural area where there is a comprehensive cancer center, but PARP inhibitors are not available. A woman has to travel 2 hours in either direction. And a lot of the women in our area cannot and never will be able to receive that kind of care. They just won't. So I don't think PARP inhibitors are going to make that big of a difference in access.
Well, yeah, to go with that, I mean, are we supposed to answer this as in the perfect world or in reality? Because in reality, no, I agree with you. The patient represent, no offense to the patient represent, we don't have women of color here today. And I have a feeling that it's because of the things you just spoke of, is we know higher poverty rates, less insurance, access isn't the same. Potentially, could this reduce disparities because it's better than chemo and having to go? Yes, but I don't know if it currently is. I know I'm a pain.
Well, also,
very true statement.
I also want to send a signal to the panel and others. We've obviously thought about this a fair amount. One thing we don't want to send as a signal in any way is that if a drug doesn't tick all these boxes, there's something wrong with it. These are possible other benefits that may or may not, and the fact if they don't, then it's not the drug's fault, or it's not, but it's helpful to kind of bring the richness of some of these discussions, and some of this will fold over into the roundtable as well.
I have a suggestion along
Yes,
that line,
Rachel.
which is
Rena, sorry.
Right now, the responses are essentially yes or no, but I think they're more nuanced because we don't have the evidence to really affirm or deny some of these questions, right?
I think you left an uncertain answer.
Well, we were going to do that, but we actually, in terms of the voting options.
They're forcing us to yes.
Yeah.
So we'll
Say
work
a lot
on that.
of
Work on
a lot
that
of
as
use
an
after,
option.
right?
We've done different voting modes in terms of level of certainty and different scales. It actually doesn't turn out to help that much. So I think sometimes almost like a yes or no adequate evidence. Sometimes it's
Clarification question, Susan, following up on your point, because we actually heard, I think during some of the patient comments, that PARP inhibitors, you suggested they might only be available in large academic medical centers, but also if you don't have to be driving back and forth all the time, that is something that sounds like it would be a significant benefit to women in more rural settings. Do you think that's still true, even if the initial point of access is more difficult to achieve?
I think it's the initial point of access that is the problem. If they can get into it, if they can make that 2-hour trip to be seen in a major academic center and get on a PARP inhibitor, certainly it would reduce the burden overall of back and forth
As
travel.
compared to women on chemotherapy.
Right.
Okay, thank you.
In our packet, it also says that there is a requisite BRCA testing to be on Rucaparib and
Rucaparib,
yeah. Is that? But it's been approved for both.
No, rucaparib is only approved for somatic and germline mutation patients.
So that is a
It
required.
requires an FDA-approved test, but in reality, any test that probably came from a CLIA lab is going to be okay. But that level of testing, as our panelists pointed out, is low in rural communities, basically because they didn't have a historic need to test or desire to test, or the resources to test.
Is it just blood? What is
blood or a
involved?
cheek swab for the germline, or you can send a piece of the tumor off for the somatic testing. It's expensive, is the other part, so there's some barriers, and unless there's an oncologist that knows about this, if they've only had one patient a year they're treating, the problem, we're dealing with a rare disease, and rare diseases are even harder in a rural community.
Yes, Don.
The second question I just want to be sure about or the statement is complexity of treatment. Is that what is intended by that?
Yes.
As opposed to, for example, the family or caregiver burden, which is a complex.
There's some overlap there. But
some overlap?
Yeah, the idea is that in some cases we'll be looking at data from clinical trials where patients have been managed to get a very rigorous treatment kind of protocol, and it could be that in the real world we'll all expect that to be very hard to match. Whereas some other drug might have a simpler protocol, but if you actually look at the clinical trial data, they would look the same, but you could make a pretty good real world assessment, especially if there is real world evidence that the more complex or the simpler regime, if you will have better real world outcomes.
So then can I ask, well, actually, I forgot, is it, Susan, you're the one that's gotten this treatment?
No, she.
Betsy, I'm sorry. Is it less complex?
Well, I think I explained before that I have a port. Most people who are getting this have a port that has to be flushed.
Right.
And they get their CA-125 tested.
Right.
And I just have it at the same time that I get monitoring for the olaparib.
Yeah.
So it's no more complex.
Okay.
In fact, I think it's less complex. And the reason is, okay, if you're having to travel for IV chemotherapy, even if you're going 15 minutes down the road, you likely have to have somebody with you to drive you both ways.
Yeah.
Because you're on other drugs that affect your abilities to do a lot of things.
Yeah.
You're also going to have to go back to that center to get a blood draw at some point. Whereas when you're getting a PARP inhibitor, you're likely going in to get your prescription filled, get your blood drawn all at the same time. And you can do that yourself. You don't have to have somebody go with you.
I remember my mother mixing the ampules of 5-FU in her orange juice as opposed to getting an IV. So those were the days.
Another one of these, at least the research team it does not feel is likely to be a major issue is the quality as measured here not capturing direct patient health benefits. There are a lot of these are kind of indirect benefits, if you will, but as far as the quality's role in trying to capture quality of life, this is not known to be an area where it would be particularly difficult to do that.
I guess I had a question to the other panelists, or how this is our first time to kind of try to process through these questions as a group. And on item C, which the intervention will reduce important health disparities across racial, ethnic, gender categories. So when I read that question, I look at this particular condition that only affects women. So it's only my mom, and it's only my aunts, and it's only my daughters. And I'm curious how, our patient partner, said, well, this is the one she didn't see that way. And I kind of had like, oh, this was one to me that was more directly potentially anyway, a health disparity issue in terms of a gender. Yeah, for sure. So I'm curious, how other people read that and how you reflect on that as it pertains to, kind of checking the box, so to speak, on your thoughts about it.
Yeah, I'm struggling with this question as well, and, by definition, where the disease essentially only affects women, is the answer yes by definition? I think in some sense, no. I think Ryan is right to express concerns about racial disparities. I personally also have socioeconomic concerns, right? Is this exacerbating women with good private insurance may be able to access these? There may be some roadblocks that Medicaid is putting up. I don't have a sense, but it seemed like no from the report, but I don't really know. Right, so I worry that the answer is yes to some of these categories, no to others. For me, I'll just reveal what I'm probably thinking I will say is that where women are so underrepresented in clinical trials generally, where there are fewer drugs which are developed targeted at diseases that primarily affect women, I feel more comfortable placing greater weight on that particular category, but that may not always be the case.
I appreciate your comment. I appreciate your thoughts on that.
All right, we've got another set of these types of questions to come, so.
You mentioned adding in another foil.
Yes, please.
And I would say what my patients often want to be on olaparib is when it's winter and they're traveling to Florida. So it gives them autonomy, ability to travel, and vacation. Because they're not tied to me, they're tied to a lab somewhere, but they're not tied to coming to see me. And it really does, that's one of these benefits that is hard to quantify, but it's definitely there.
Yeah. Again, we'll be evolving the language on this as we learn and go forward. So that fits somewhat in the reduced complexity. And I would want people to consider it there. But our language isn't perfect for that very good example.
Are we going to get to a different screen where it allows us to fill in for G?
If you have a G right now, why don't you?
I have a G.
You have a G. What would you like to suggest?
Well, I think that this value of potential long duration responders or super responders is something that is not necessary. The quality may not be potentially a good measure of, and or at least it's not. At least the literature in the economics field is actually has some debate about that, and so I think that that's a standalone thing that we should consider. Actually, for all oncology products.
Right. Okay. Well, I might want to take that under advisement because I think you could include that as you're thinking around A, if you feel that that's a direct patient health benefit that's not captured. Otherwise, the challenge here is that we've heard about. I just want to note we've heard about this, but there are no data.
Right. Exactly. I mean, exactly. There are no data, but I mean, it's a point of contention in the economics field.
Oh, absolutely.
Whether this
the distribution
bifurcated survival curves are not really captured in our method in the way that we do that, or that we do it in the sensitivity analysis, but that's not really the main result. And so I'm just saying that could be something that we should consider.
So I'm going to suggest, let me take that and see if you agree with my suggestion, which is, to the extent that you believe that effect that was talked about before, where we're seeing a median result, but that may not capture that there's a large or relatively large tail end of patients who are living much longer. That to the extent that that's not captured in the cost effectiveness and the quality measurements here, that you should include that as a factor under A.
I mean, I think our colleagues who ran this analysis may be able to comment more about this, but I think
I was just telling Dan, if we want to directly model that scenario, we're actually running into this with our next review, but there is a way to directly model what you're just stating, and that's something like a decision tree up front, which predicts responders. You can have sensitivity around those responders to see if there's a lower and an upper confidence interval. Then we move them to a separate model, which incorporates the quality. So there is a direct way to model it, but I think it is a really good point. How much weight, in a sense, do you place on the responder, and how well connected is that to progression-free or some type of remission outcome? So it's a good point.
I mean, just from first principles economic thinking, this value of subsequent innovation and the value of hope that is attached to this durable response or the random draw that I happen to be the person who is the super responder in the oncology setting is a real separate category and one that should be considered.
Great point.
All right, so for now, let's consider that under A, and we will, I think we'll have to do that. And I seem to have hit a button that I didn't even know existed, and we're starting to see the results. But one other person can enter their response, and then we'll be done. Oh.
Can I just say?
Choose your vote again.
Okay.
It seems to me that what Rena said.
Sorry, can I ask you to pull the microphone just so the people on the webcast?
What Rena said relates somehow to what Shumei, if I pronounced that right, said, that you don't know whether the number of prior chemo lines makes a difference in PFS or overall survival, or whether it's something else that creates the extraordinary responder. I think it has to do with your immune system somehow. But these are, again, questions that float in the air and have not been answered and certainly not quantified.
Okay, could you please vote again? Sorry, just those of you who haven't voted, vote again, so I won't touch anything until we get to that. Step away. Okay, thank you. All right. 5, significant direct benefits not adequately captured. You can read this as quickly as I can probably. 7 out of 9, reduced complexity. 3 on the disparities. 8 on the reducing caregiver and family burden. 7, novel mechanism. And 5, significant impact on work and overall productivity. All right, given the time and the amount we want to be able to allocate to the policy roundtable, let me move quickly to the next set. These are called contextual considerations. Sometimes it's hard to understand why these wouldn't sometimes be viewed as a benefit versus context. Try not to let that worry you. It's all kind of going to be captured as part of the report. But here's the way the question is worded. Are any of the following contextual considerations important in assessing PARP's long-term value for money in patients with recurrent BRCA mutated disease? Please select all that apply.
I can only answer 2, and it logs me.
Oh, they have to reset.
Oh, okay.
Steve, could I just ask a quick question while I bring that up? When we use the phrase significant uncertainty in the last 2, is it reasonable for me to interpret that as, or substitute, there is questionable certainty? And the reason I say that is because I'm just, by my nature, concerned about the word significant from the standpoint of quantitative versus scientific.
Substantial help you?
Substantial certainty?
uncertainty.
Substantial uncertainty.
So what you're being asked here basically is that if you are advising, let's say you're advising a payer who's going to be judging value, or a clinical group, what do they need to think about in terms of the context that you think is important to thinking about the value of these innovations? So, is this a particularly severe condition? Is that important for people to think about? Do you think there's a high lifetime burden of illness? What that means, again, is not just something that's acute, but something that might be chronic, over decades even. So that there's an accumulated experience of having a challenge with your health. Is that important in the context of thinking about these treatments? Here are we talking about the first treatment ever to offer any improvement? I'll just jump out and say no, right? I mean, this is there for the unusual situation where we have, let's say, a rare genetic disease that's never had any treatment, and we're here to talk about the first treatment for this condition that's ever been available. This is maybe an advance, obviously, but it's not the first treatment ever for patients with ovarian cancer. The other two are more around kind of, how do we think about the relative certainty we have around long-term side effects and benefits?
Yeah, and my question was on the last two.
Mm-hmm.
But you don't want the C to be any additional.
That's correct.
Right.
Hopefully we've covered that with the net health benefit. All right. Any discussion, question about these? You guys, again, can feel free to ask each other, give your ideas, ask the panel. This may feel a little bit more straightforward. In other cases, it may not. We know that we're not giving you an anchor, so we say a condition of high severity. Are we asking you to compare that to toenail fungus or to some other cancer? This is basically thinking about the big picture. So it's basically everything. Everything that would come before judgments about value in healthcare. That didn't help very much, but that's the general goal.
And this
okay.
Just for recurrent BRCA mutated disease.
Correct. All right, one more response, and then we'll be ready to show.
So I have a G for an F, I guess, which is about this financial toxicity issue. Because clearly this kind of oral versus IV therapy issue has very significant access concerns, particularly for folks who are commercially insured and who may not have catastrophic coverage in the medical benefit drugs at all. Have you, I guess the previous question around caregiver broader, I guess the disparity issue maybe addresses
Let's
that.
think about how we might weave that in going forward. I'm not sure it fits as a contextual issue in some way. It's a very practical issue, and I'm just trying to figure out. But let's think about that. I'm not able to quickly identify that as kind of a generic contextual issue that would be easy to frame for this and other conditions. All right, we've got all 9. There we go. Okay. 8 out of 9, care of individuals with a condition of high severity. 6, high lifetime burden of illness. 1, first to offer any improvement. 2, compare there's a significant uncertainty about long-term risk of serious side effects. And only 4 out of 9 also said that there's a significant uncertainty about the durability of long-term benefits. Okay. We are all going to debrief on this and learn from the experience. We also want to hear from stakeholders, going forward about how this does or does not add to the transparency and, the tangibility of some of these issues and discussions. But now what we're going to do is move straight to, in a sense, the summary vote on value. So remember, ICER's conceptual value framework says that what we just talked about, everything around comparative clinical effectiveness, other benefits, contextual issues, and the incremental cost effectiveness results are now placed into the blender, and we ask you a vote on value. So given the available evidence on comparative clinical effectiveness and incremental cost effectiveness, and considering other benefits and contextual considerations, here's a specific patient subgroup. In patients with recurrent BRCA mutated disease, what is the long-term value for money of olaparib compared with PLD plus C chemotherapy? The reason this is the value vote that's being taken is, again, according to ICER's methods. This is the one where the cost effectiveness range was close enough to kind of a zone where a vote will be taken. If it's too high or too low, we don't vote. When it's kind of in a middle range is when the votes are taken. So having just gone through that exercise of thinking about very concretely and voting on the other benefits, can I just invite Brett to remind us what the cost effectiveness result was?
Yeah. So you'll see page 15, the very first bullet is the population and comparison in question, along with the 3 sub-bullets, 2 of which label the limitations, which I've discussed previously. First, this is not a direct comparison. This is historical data. And second, we have that combination of BRCA mutated and non-BRCA mutated patients. You'll also see on page 16 that the top rows of both tables show the cost effectiveness of approximately $80,000 per life year gained and $145,000 per quality adjusted life year gained approximately. You'll also see on the left-hand side the intervention and non-intervention costs, and on the far right-hand side those effectiveness estimates.
Any questions about the cost effectiveness results? Okay.
You might want to just remind folks of the standards that we've used in the past in terms of ranges, or?
Well, the range that ICER uses for value-based price benchmark is between $100,000 and $150,000 per quality. Some health economists believe thinking about lower ranges makes sense for some situations, higher for other. But in general, we usually present results at 50, 100, and 150, partly for historical reasons, but that's the range within which we vote. All right, so again, your job is to help integrate all of these considerations, and before you vote, I'd like you to make sure that you hear other people's thinking here. Can I invite someone who feels confident that they know what they want to vote, express their reasoning? Everybody's so confident that they vote. Jump right out of it. Rena, do you know what you're going to vote?
Yes.
Tell us what it is and why.
I'm going to vote intermediate.
Because?
Because while I'm reasonably unconvinced of the intrinsic value of these drugs in the setting, in the FDA-approved settings that we've discussed, I do believe in the option value of this therapy and in the potential value of this therapy moving forward. And so I worry that a focus simply on the intrinsic value as we have in these analyses now would misunderstand my acceptance that this is probably has some very important clinical value moving forward.
Okay. That was very well said. I also want to help people with the terminology. Again, the word value can be helpful and obscuring at the same time. So here, value, long-term value for money explicitly asks you to remember to weigh what you see as the clinical benefits, the net clinical benefits, and all these other benefits and things, and the costs. So it is a judgment of that long-term value for money. So you're not necessarily saying that there is no clinical value or that there aren't other benefits. It's part of that balance. Anybody else have a strong sense? Oh, I should also just, I'm sorry, a little bit of guidance. For those of you who voted that you did not think the evidence was adequate to demonstrate its comparative, its net health benefit, you would generally, the usual approach is to think of that as you would probably be at low value. If you don't think the evidence is there to say that it's better clinically, it's more expensive. But again, we encourage you to also think of this as the bigger picture. Even if you're on the borderline about the evidence being adequate there and you would have said low value, perhaps these other benefits or contextual issues are something that you want to weigh. Anybody else have a pretty clear view of where they're going to vote?
And this is towards the upper end of it's at 146,
The
right?
base, at what's called the base case analysis, that's true.
Unless the committee compels our pharmaceutical colleagues to lower the cost, which would be great, right? Oh, we did. No response from that.
The policy roundtable is coming up.
One reason I asked about their commitment to financial access is for exactly this reason. So if you're offering the value of hope with not a lot of intrinsic value, but you're giving it away to patients who, may benefit, then that's a different calculus than what we are living with.
Is it reasonable? I'm not going to look at Brett. Is it reasonable to factor in non-econometric value for money guesses based upon some of the testimony we've heard today, or is this econometric value?
No, I think I've tried to say that the cost effectiveness is one important part of a judgment of value. So are these other pieces. Yeah. And yet there is I can't tell you 1.2 times your vote on other benefits equals some cost effectiveness to put it in balance. That's the challenge of doing this, and it's part of why we do it in public, and we learn from each other, and we share the uncertainty, but also try to wrestle with that tough question. And people will feel differently about it. That's part of what we're doing. So I'd be interested, is somebody going to vote high value who would like to judge to present why? Or is somebody going to present a low value vote and suggest why? All right, I'm putting you on. Okay, Jill?
I'm voting low value because in this setting with a single arm trial, one would hope that at $146,000 per quality adjusted life year, there would be better certainty on what you're getting for your money. And so my hope is that in doing that, it will put some pressure on pharma to be more reasonable.
Some international systems of health technology assessment do think of uncertainty in that way, that if you have a certain degree of uncertainty, you wouldn't, if you have the more uncertainty, the lower you would want your cost effectiveness to kind of balance that out. We don't do that mathematically, but that line of reasoning is certainly one that's quite rational, some people would say, and has been used before. Okay, why don't we go ahead and vote? 3 more people, please. Everybody press your vote one more time. Make sure it went through. There we go. All right. 4 intermediate and 5 low value. All right. Well, that was a very rich discussion around these issues. Sometimes when you get a split vote, it means one thing, and sometimes it means another. Here, I think it means that we've wrestled quite openly with some of the difficult issues around the balance between uncertainty, the kind of other benefits that these drugs potentially offer patients, et cetera. So thanks a lot for your votes. So what we're going to do is take a 2-minute stand-up break to kind of constitute the panel here for the policy roundtable. It looks like it's going to be 3 minutes because I see CEPAC members who need to take a rest break. So we'll reconvene in 3 minutes. AKA stand-up panel. That's right. That's right.
Sure. I really enjoy your comments.
I really enjoy it.